Good afternoon, everyone. Happy World Kidney Day. Hopefully you remember to put on your most colorful socks to celebrate the Suck It to Kidney Disease campaign and take a picture, share it on social media. And if you have time to stay after the conference today, there'll be a group picture as well. All right, so thank you for coming again. This is a very special seminar today. Some of you who pay attention to emails might have noticed that we were originally scheduled to have a different speaker who couldn't give the talk today, and then Pinelopi came and rescued us after I asked her at a very last minute to give this on a very special day. So I'll say a little bit about her and then let her present on her research. So Dr. Kapitsinou is an associate professor of medicine in the Division of Nephrology here, and she received her education, medical education in Greece. It was very interesting for me to find out that she, I think, graduated as the #1 medical student in her class of 500 in the medical school in I'm not going to be able to pronounce it. National Kapodistrian University of Athens received the top 1% of medical students at that medical school award, in addition to many other awards. She then moved to the United States and did her residency at Jacobi Medical Center, her fellowship at UPenn, and the research fellowship at Vanderbilt, so all very prestigious institutions. Before coming to Northwestern, she established an independent laboratory at the University of Kansas Medical Center, and then moved to Northwestern in 2019, right before the pandemic, which is a great time to move and start up a basic science lab. Her research focuses on the role of hypoxic signaling in kidney disease with the goal of leveraging adaptive and maladaptive responses for therapeutic development. Her research is supported by NIH, and the lab uses very integrative approaches, relying on genetic, biochemical, and cell biological methods, both in vivo and in vitro. This very creative work resulted in many high-impact publications, including a very recent JCI, Journal of Clinical Investigation publication, for which she was actually interviewed in the news media here at Northwestern. So if you go to her faculty profile, you can read a little bit about that paper, and you could actually go and read the original paper, and I'm sure she'll share the work on that project today. For all of her accomplishments in this area, she has generated national and international impact. I'm just going to highlight a couple of things. So she sits on the Scientific Advisory Board for GSK, the Deprivation Steering Committee. And because of her accomplishments, she also was asked to be a standing member of NIH Study Section. She's an editorial member of AGP Renal Physiology, Frontiers in Cell and Developmental Biology. And on top of all of this, she also is very committed to mentorship. There's more than 20 mentees listed in her CV, and several of the mentees are people you might recognize. Some of you might remember Boaz Bishop, who graduated from the transplant program here. Maggie Fain, who's I think in the audience, counts her as one of her mentors, and Dr. Kapitsinou also sits on the committees for Esther Liu and Johnson Yang, who I think I also saw here today. And she also contributes to the faculty development for young investigators, both in adult and pediatric nephrology. So some of you might know Vidi Dalal and Shana Bradford, who joined us earlier this year. Pinelopi also finds time to do lots of service. She is co-directing the biomedical resource core of our O'Brien National Research Center, and sits on a number of committees. I'll just mention the American Society of Nephrology Scientific Program Committee and the American Heart Association Scientific and Clinical Education Lifelong Learning Committee. So anyway, Pinelopi, thank you very much for agreeing to do this on short notice and on a very special day.
Thank you so much, Tamara, for this super kind and generous introduction. It's really a great pleasure and honor to present on a very special day today. World Kidney Day. It's a great reminder of how important is our mission in patient care, research, and education. Today I would like to give you some highlights of work that we have been doing for some time now. And as Tamara already, described, we are interested on how hypoxia responses affect kidney in different disease settings and how actually we can exploit this biology and find new therapeutics for our patients. So in outline, we are going to discuss about the oxygen sensing machinery and potential therapeutic implications, and then I'm going to focus on our recent work on endothelial PHTs and glycolysis, how it regulates kidney injury, and then I'm going to close with the role of endothelial PHT3 in AKI to CKD transition. So I would like to start with a clinical case, for our fellows. It's actually a very familiar bread and butter case. 62-year-old male with history of hypertension, type 2 diabetes, comes to the emergency department with fatigue, reduced urine output, swelling. He apparently was taking ibuprofen for the last few days, and he also has been on antibiotics for a UTI. His vital signs are stable. He does have bilateral lower extremity edema. And then he has this serum creatinine of 3.6, while our records show that 6 months ago, his kidney function was close to normal with a creatinine of 1.1. The rest of his labs showed normal electrolytes, a urine analysis with muddy brown granular casts, some proteinuria, and a serum lactate of 3.4. We did a renal ultrasound, which showed normal-sized kidneys and no evidence of obstruction. So what therapies do we have? Any fellow would like to volunteer? Do we have a magic bullet to help this patient go back to where it was? Okay, silence. And, this is actually reflects, the reality. We actually don't have effective treatments to help this patient recover his kidney function. The diagnosis here, acute tubular necrosis secondary to ischemia and NSAIDs. The patient received supportive care with diuretics and discontinuation of nephrotoxic medications. So now how this patient is going to evolve? Again, it's not clear. There are different scenarios. One scenario could be that he recovers very close to where he was before. Another scenario, and this is not the only one, could be that his kidney function actually does not have a remarkable improvement, and he stabilizes in this level of a creatinine of 2.8 and has some persistent proteinuria. And then 6 months later, we give him the diagnosis of stage 4 kidney disease, and he's on a way, really preparing him for, future treatments that involve dialysis replacement therapy. So really, the core of our interests is how this kidney that has been in the syndrome of acute kidney injury, how is responding to different insults. And, as you can see here, the kidney is a very heterogeneous, let me basically see here, does this work? Yeah, okay. So it's a very heterogeneous organ with different cell types, and this is the normal structure here with tubular cells, endothelial cells, macrophages, pericytes. So this is what we see under normal conditions, but in the condition of AKI, there is this injury with necrotic apoptotic tubular cells, injured endothelial cells, inflamed macrophages, really a chaotic situation. And this injury, if it resolves successfully, we have an outcome of complete repair, but we now realize that this injured situation may result in maladaptive repair and a fibrosis, tubular atrophy, persistent inflammation, phenotype. And this trajectory from kidney injury to maladaptive repair can be affected by coexisting conditions like aging, diabetes. In the past, back in the 80s, people thought that AKI is a benign condition that results in repair and recovery. But now it has been very well established that AKI in up to 30–40% of cases can actually result in maladaptive repair. And why this is happening? Well, kidney is actually performing really some sophisticated energy-requiring functions, and this happens in a very tight oxygen supply. So you can see here that the tissue oxygen is obviously determined by a balance between oxygen delivery, therefore renal blood flow, and oxygen consumption. But a lot of clinical conditions that we see really affect renal blood flow, and we have patients with intravascular volume depletion, sepsis, hepatorenal syndrome, medications, where renal blood flow is compromised. And now you have an organ with significant oxygen consumption, reduced delivery. Well, this imbalance results in hypoxia. So, it has been in our field a huge theme of how we can come up with effective cytoprotective approaches. Approaches in which, when there is a predicted insult, and this predicted insult can be, for example, a cardiac surgery, what type of approaches we can come up that can protect the kidney, increase the resilience, and result in no AKI. So inspired by this theme, there has been a lot of basic science research trying to understand and figure out what are potential effective cytoprotective approaches. And one major approach has been this ischemic hypoxic preconditioning that has been really inspired by the principle of hormesis, where you have beneficial effects of a treatment that when it's delivered at a higher density is actually harmful. So in ischemic hypoxic preconditioning, now more than 20 years ago, Joe Boventre showed that one or several episodes of brief ischemia hypoxia followed by reperfusion can actually generate profound protection against a subsequent prolonged episode of ischemic injury. So this this concept of ischemic hypoxic preconditioning has actually been shown that it delivers protective effects not only in the kidney, but in various organs, the heart, the brain, the lungs, intestines, liver. It works, it's preserved in different species, but how about translation in humans? Well, there have been, different studies in this issue with conflicting results, I will say. So I have shown here a study from New England, it's now, 10 years ago, in which the investigators studied the effect of remote ischemic preconditioning on outcomes of cardiac surgery. And here, this remote ischemic preconditioning was accomplished by a blood pressure cuff, which delivered ischemia by inflating the cuff at high blood pressure, I think more than 200, 4 cycles of 5 minutes of limb ischemia, following by reperfusion. But the results of this study were really negative. At that time, the potential reasons for failure that were mentioned were comorbidities, old patients, concomitant medications that could have affected, the ischemic, the response to the ischemic insult. But I think you can appreciate here that this is not really a modern approach to treat patients and not like really precise this, remote ischemic preconditioning. So do we have actually a more precise pharmacologic approach to deliver ischemia? Well, exciting advances in the field of oxygen biology have actually revealed a very sophisticated oxygen sensing machinery that regulates responses to ischemia and hypoxia. And this actually resulted in a Nobel Prize for, these three investigators back in 2019. This is Peter Radcliffe, who is actually, in Oxford, and he's a nephrologist. Bill Kylen, to be complete, Greg Semenza. So let me navigate you through this hypoxia sensing machinery. So a central component is the executors of the hypoxia responses. So hypoxia-inducible factors are transcription factors that really are constantly made, but under normoxic conditions, they are degraded. However, under hypoxic conditions, they are stabilized, they translocate to the nucleus, and this HIF-alpha subunit dimerizes with ARNT, which is the HIF-beta constitutive subunit, and forms this complex which binds to the hypoxia-responsive element of genes that respond to hypoxia. And this binding results in activation of a big transcriptional program that serves the purpose of adaptation to hypoxia. So, one major example here is EPO. So HIFs, and especially the HIF-2 isoform, upregulates kidney EPO synthesis. And by promoting EPO synthesis, it promotes the erythropoietic potential. You have more red blood cells, more oxygen delivery, aiming to alleviate the hypoxia. Now, you can appreciate that in top of EPO, there are, angiogenesis genes, metabolic genes, genes involved in proliferation, and really here the goal is to prepare or deliver a rapid response to reduce the oxygen supply. Now, I want to highlight that this pathway is already being exploited in the clinical arena. So there are non-selective PHT inhibitors that have been approved for renal anemia, and we have them. Actually, we are started to see them even in the dialysis units here in the U.S. The first approval came in China. It followed by countries in Japan, European Union, and then the FDA approved the first agent back in 2023. Therefore, patients, with dialysis, requiring kidney disease, are exposed to these PHT inhibitors. Now, beyond the renal anemia, which is, we use the PHT inhibitors because they induce EPO, the next clinical application is actually ischemic disorders. We have, so as a postdoc, like my main project was actually how to use this biology to improve outcomes in AKI. And you can see here, back in 2014, we published that when you pre-treat an animal with this PHT inhibitor prior to AKI, you have improved outcomes with less, kidney injury, less, injury markers, reduced inflammation. And this, was along with the work of other labs, as you can see here. Different labs have published, very similar results. So when you activate HIF, even by different means, like different PHT inhibitors or, other compounds that interfere with the activities of proyl hydroxylases, you have protection, against AKI. And the mechanisms that, really involve this cytoprotection are diverse. So I have highlighted here metabolic reprogramming, suppression of inflammatory responses, beneficial responses that reduce maladaptive oxidative stress, anti-apoptotic effects, cytoprotective factors, and so on. However, the real-world problem that we are facing now is that we know that, patients don't really give us warning when they are going to have an AKI episode. They come in the hospital, they have an established injury, so they are like in the context of ischemic injury, they may be on a reperfusion state. So is this biology relevant? Can we harness it to improve the outcomes of patients with established AKI or when they are on this AKI to chronic kidney disease transition? Well, it turns out that the results when, people have used these PHT inhibitors after injury have been very conflicting. And why is that? Well, the injured kidney is a very complicated, chaotic environment. And, as you can see here, there is mitochondrial dysfunction. Actually, these tissues, they may already have HIF stabilization. There is oxidative stress, metabolic dysregulation. And the injured kidney is a kidney that, may have already, other conditions because of pre-existing kidney disease, like glomerular injury, atherosclerosis, and so on. So in this environment, these, preclinical approaches have not worked out well. So, for the last 5-6 years, we have been trying to understand on a very precise approach how we can, really harness the hypoxia responses in a cell-specific manner to alter this chaotic microenvironment. And, we have been actually very interested in endothelial cells. So why endothelial cells? Well, there are several reasons, but, here I show you that the hypoxia pathway is very important for endothelial cells because in response to hypoxia, we have angiogenesis. And we now know from work of, Peter Carmelier that the way that the endothelial cells have this angiogenic response really involves metabolic reprogramming. So really the endothelial cells change their metabolism in order to be able to adapt and make, more vessels and deliver oxygen. However, there is a paradox. The fibrotic and hypoxic kidneys, they are actually characterized by capillary rarefaction. And we know that this reduced capillary density is actually a very good prognostic factor for kidney disease staging and progression, similar to the tubulointerstitial fibrosis. So key questions that we have been trying to address in the lab is how endothelial oxygen sensing affects endothelial cell functions. In the setting of injury, how endothelial cell metabolism is affected. What is the impact of endothelial HIF activation through PHT inhibition on kidney fibrosis? Trying to answer these questions, we have used different approaches. So I highlight here our work published, now 3 years ago, where we used mainly an in vitro approach with a chemical inhibition of oxygen sensing proyl hydroxylases to see the outcome on different endothelial cell functions. And what we found is that when endothelial cells are subjected to a pan-PHT inhibitor, which in this case is the DMOC, they actually have a hypoproliferative response, reduced migration capacity, reduced angiogenic abilities. So these are assays that, show how endothelial cells form tubes. And you can see here that the cells that are treated with the inhibitor, they don't do actually very well. This is an assay that measures the migratory capacity of these endothelial cells. And, PHT inhibition had a negative effect. So this was actually a surprise to the field, but it has been reproduced by different studies. So, I have included here a study where they used not a pharmacologic approach, but a genetic approach to inhibit the proyl hydroxylases, and they saw also a hypoproliferative response on endothelial cells. Nevertheless, the in vitro studies do not necessarily recapitulate what happens in vivo. So, we moved to in vivo studies, and first we decided to use the CRE-LOX-P approach to inactivate the main oxygen sensor, which is proyl hydroxylase 2, PHD2. For that purpose, we used mice with an inducible endothelial CRE and a conditional allele for PHD2. So we generated these mice. We subjected them first to ischemia reperfusion injury, and then we induced deletion of PHD2 specifically in endothelial cells using tamoxifen, which allows the activation of this transgene. And to our surprise, what we found was that when we deleted the main oxygen sensor, PHD2 on endothelial cells, there was not a difference in the outcome of these mice. So you can see here, mice that lack PHD2 in endothelial cells were pretty similar to the CRE-negative controls. This was done by different metrics, injury score, picroserous staining, and the expression levels of different profibrotic genes. However, we did notice that endothelial PHD2 deletion did not actually affect HIF-1 protein levels in the kidney, while there was, stabilization, in different tissues, as you can see here from our previous work in the lungs. So it seemed that different vascular beds had different sensitivity to PHD2 inhibition, with the lung, for example, being very responsive, showing stabilization of HIF-1 and HIF-2, whereas the kidney did not show a response. So with these results, we decided to look at the expression and the function of other PHD isoforms. And for to this end, we analyzed single-cell data publicly available from mice and humans. And what we found was that in addition to PHD2, PHD1 and PHD3 were significantly expressed in different endothelial cells in the mice, but also in humans. So this expression pattern of these PHD isoforms in the mouse and in the human really suggested that oxygen sensing in endothelial cells can be complicated and not be mediated by the main oxygen sensor PHD2. PHD1 and PHD3 isoforms may have important roles. So because of this, we decided to really delete all the proyl hydroxylases in endothelial cells. So for that purpose, we used a mouse with conditional alleles for the 3 isoforms, and we deleted through a CDH5 CRE-ER the different PHD isoforms, and we generated this mouse that has a super hypoxic response in endothelial cells. And as you can see here, by Western, there is stabilization of HIF-1 and HIF-2 in the kidneys of these mice. And also, by immunofluorescence, we could validate the deletion of the different, isoforms in endothelial cells, as you can see here in the mutants, compared to controls. Now, under baseline conditions, 2 weeks after the recombination, these mice did not have any obvious kidney phenotype. But when we subjected them to injury, we found that they had more AKI to CKD transition. So as you can see here, at day 14, post the initial unilateral IRI, mice lacking the 3 proyl hydroxylases, they had more injury, more fibrosis, and significantly higher expression of profibrotic genes like LOX-like2, TGF-beta, ACTA2. Along with that, there was more pronounced capillary rarefaction, as you can see here by endomucin staining. And when we did a bilateral ischemia reperfusion injury model, we found also that these mutants at day 14, they had increased BUN and reduced GFR. So overall, now on this model, we see that it recapitulates all the key aspects of AKI to CKD transition with more injury, fibrosis, capillary rarefaction. So to better understand how endothelial cells modulate this environment, we took advantage of modern, technologies offered now, for example, through single-cell RNA-seq. And when we subjected our mutant mice compared to controls to single-cell RNA-seq, we could see that different cell types, they were showing prominent maladaptive responses. So you can see here that the tubular cells of these endothelial mutants, they were showing enhanced TNF-alpha signaling, enhanced apoptosis in distal tubules, enhanced pro-inflammatory responses in the collecting duct. Since our genetic approach affected endothelial cells, the responses in the tubular cells are really responsive to what happens due to alterations in endothelial cells. So we focused more on the endothelial cells to better understand how they are modulated by our genetic approach. And what we could see is that we had 3 different clusters of endothelial cells: cortical, medullary, and endothelial cells showing signs of dedifferentiation. And the endothelial cells that were more actually responsive to our approach were the medullary renal endothelial cells, cells that live in the medullary environment characterized by more pronounced hypoxia and more, hostile environment because of high osmolarity. So as you can see here, these endothelial cells, they show enrichment for hypoxia and enrichment for glycolysis. And in fact, all the glycolytic genes were significantly upregulated in the endothelial, medullary endothelial cells of these mutants compared to control. So really, this actually data validated that our causal metabolic approach worked, that we had hypoxia in endothelial cells, we had increased glycolysis, and apparently this response was not beneficial because we had more AKI to CKD transition. We focused on this glycolytic response, and we wanted to assess, is this glycolytic response really causally linked to the maladaptive outcomes that these mice developed? So, for that, we assessed more the tissue microenvironment, and as you can see here, we could see more inflammation in the mutants with increased leukocyte migration, inflammatory markers like CXCL12, MIF, CD74 were significantly increased in the mutants, and this response actually was associated with significant increase in macrophages in these post-ischemic kidneys. So we could see by analyzing how endothelial cells interact with macrophages some very interesting interactions. So you can see here, for example, the increased CXCL12 in endothelial cells is really, predicted to respond to interact with CXCR4, in macrophages. So, really this data generated new hypotheses for how enriched interaction of endothelial cells with macrophages can promote a maladaptive, inflamed microenvironment in these tissues. So far what I have shown you that when we target the hypoxia response in endothelial cells, we activate HIF through PHT inhibition, we have increased glycolysis, increased inflammation, and fibrosis. So what is the role of HIF in this signaling? Well, to address that, we generated mice that lack the HIF-beta subunit, ARND, which paralyzes the hypoxic response in endothelial cells. And we repeated our AKI to CKD transition and model. And what we found was that these actually mice, they show less injury, less fibrosis, and less reduced expression of profibrotic genes when they are subjected to unilateral IRI, really showing that the activation of endothelial HIF is actually playing a crucial role in this model. Now, how HIF is doing that? So to understand that, we started to look more on publicly available data. And here, we took advantage of kidney biopsies, or actually not biopsies, let me correct, autopsies from patients who had severe AKI and were hospitalized in the ICU. These patients unfortunately passed away, and the kidneys were used for single-cell RNA-seq. And we extracted the endothelial cells from this set, and what we could see is that the two pathways being more, upregulated was interferon gamma signaling and hypoxia. So really, showing that what we see in our mouse studies is very relevant to human AKI. And we looked also on, endothelial cell clusters available through KPMP data. And again, we could see upregulation of hypoxia in AKI, in CKD, very similar responses. Now, the other a key signature that we could see in these endothelial cells from human AKI is that all the glycolytic genes that I mentioned before from our mouse studies were significantly upregulated. So you can see here PGK, lactate dehydrogenase, and so on. So at this point, we wanted to assess, okay, what is really the role of enhanced glycolysis in this model? So as I said, all these genes are upregulated. How can we modulate them and see, the outcome? To better select which gene we are going actually to target, we used, a progress that has been made by really, prominent scientists in this field. So this is work, from Josh Rabinovich, who actually showed that the key enzymes that regulate glycolytic flux is GLUT, hexokinase, PFK, and then there is this transporter, MCT4, which regulates lactate export from intracellular to the extracellular environment. And by exporting lactate, it regulates glycolysis because lactate is the end product of glycolysis. So we got interested on this MCT4 because was like a druggable target, and we validated that our mutants had actually increased expression of MCT4 on endothelial cells. And then, some facts about MCT4, it belongs to the SL616 family, has different, so they have different isoforms, but MCT4 is highly expressed in hypoxic cells, like cancer cells, and is a hydrogen lactate importer. HIF-1 has been shown actually to induce MCT4, and, of interest, actually CD147 passaging, as it is a molecule that has already been implicated as being very important in inflammatory responses. It actually assists MCT4 in folding, stability, membrane expression. So to target MCT4, we actually used a compound that is called cytosincopin. How we learned about this compound? Well, there have been studies in the tumor biology where, they do different type of screenings to understand, the metabolic sensitivities of cancer cells. And they have identified that cytosincopin sensitizes cancer cells to killing by metformin. And it does this because of MCT4 inhibition. Now, this compound apparently has been in our field for some time. So, this is a study from, JAMA, published in '59, where cytosincopin was given as an antihypertensive. And the mechanism of action for, blood pressure control was by depleting catecholamines from peripheral sympathetic nerve endings. However, really its clinical use was not successful because it was very mild antihypertensive, was associated with some nightmares and so on. So really, it didn't survive. But it's a compound that has been given to humans. It targets MCT4, and we used it for our mouse studies. And what we found was that actually when we gave the compound in mice with established kidney injury, and these are these endothelial mutants, we could restore, improve kidney repair. So you can see here, this is the mutant mice following IRI treated with vehicle. They look small, shrink, fibrotic. This is the compound treated kidneys, and this is like uninjured. I think you can agree that this looks more close to the uninjured as opposed to the fibrotic. And you can see here, histology pictures of, treated mice with cytosyncompin compared to controls. There was a remarkable improvement in injury score and fibrosis. And this was reflected also at the molecular level with significant suppression of profibrotic genes, as you can see. We next wanted to assess, because the inhibitor was given systemically in mice, if it does work specifically on endothelial cells. And here you can see that we have an in vitro assay where endothelial cells are basically inflamed by the addition of IL-1 beta and hypoxia reoxygenation. But when we treat cells with cytosyncompin, we can suppress the expression of endothelial cell adhesion molecules like VCAM and ICAM. And this reduced expression of VCAM and ICAM result actually in reduced interaction between endothelial cells and macrophages at the functional level. We got similar results when we inhibit MCT4 not with cytosyncompin, but with an siRNA approach, which is specific to MCT4, as you can see here. We went ahead and started to profile the metabolic impact of cytosyncompin on endothelial cells. And what we found was that when you treat endothelial cells with cytosyncompin, you actually reduce glycolytic ATP production, as you can see here, compared to the control cells. And you see an increase in ATP derived from OXA phosphorylation. So this was done by seahorse, but also when we looked at metabolites, we could see that metabolites like alpha-ketoglutarate, fumarate, malate, which are from the TCA cycle, they are preserved in cytosyncompin-treated cells compared to controls. And now this metabolic program was associated with reduced inflamed responses on these endothelial cells. So as you can see here, we could see reduction by cytosyncompin in phospho and FKB compared to controls. And how this metabolic reprogramming results in suppression of inflammatory responses is really, work in progress right now. So, in summary for this work, what we showed was that when you target the oxygen sensors, proyl hydroxylases in endothelial cells, following ischemic AKI, you actually exacerbate maladaptive repair, resulting in more CKD picture. And this was associated with a metabolic program of increased endothelial glycolysis. And if you block this program using an MCT4 inhibitor, cytosyncompin, you can improve kidney repair. So we are very excited with this work, and right now we are doing, studies in wild-type animals, profiling, the responses, and, at the same time that we published this manuscript, there have been different studies from different fields. So as you can see here, a preprint from pulmonary fibrosis, from atherosclerosis, cardiac hypertrophy, where targeting of MCT4 delivers actually therapeutic effects with reduction of pulmonary fibrosis, a reduction of atherosclerosis, improved cardiac remodeling. We are trying now in the kidney field to see whether it works on different disease models, which cell types are affected, and who knows? Maybe it will lead to a novel therapy, for AKI to CKD. We are very excited. So now, I see that we have, like 5 more minutes. I want to show you one piece of additional work that we are doing that it's clinical relevant. And for the sake of time, I'm going to show you, something very, very specific that, I would like to emphasize. So without going into details, I will say that after this bazooka approach, where we targeted all the different proyl hydroxylases, we went back and did studies with the individual isoforms, PHD1, PHD3. And what we found was that the endothelial PHD3 actually was the isoform that was differentially regulated following AKI. Now, when we did studies with just targeting of endothelial PHD3, we could see that these mice following AKI, they recapitulate the phenotype of the triple mutants. So just deletion of PHD3 was actually sufficient to promote maladaptive kidney repair. So, I'm not going to go into details, but I will say that PHD3 inactivation in endothelial cells promoted inflammation, promoted macrophage infiltration. We have done single-cell RNA-seq, in these kidneys, and we have seen increased interferon gamma response in endothelial cells of these mice. And then we have done mechanistic studies where we have inactivated HIF in the setting of interferon gamma stimulation. And what we can see is that PHD3 inhibition actually regulates the interferon gamma pro-inflammatory genes in a HIF-dependent manner. And I want to say that, this is actually important because we are going actually to go back to how this affects, our patients. So I want here to highlight data from a study that was published, in Kidney International not long ago, just in 2023, where they showed that proyl hydroxylase inhibitors like DMOC or hypoxia actually activates the transcription of ApoL1 in tubular cells, in podocytes. That's, what was this paper, about. And, based on that, we said, wow, so PHD inhibitors, they, increase ApoL1. So obviously there is a concern because these PHD inhibitors, they are given, in patients, even on dialysis now. And the fact that these patients are already on dialysis, it doesn't mean that they are protected from ApoL1 upregulation. We know actually that these patients with ApoL1, they are very susceptible to sepsis, and this enhanced interferon gamma response can set them up for bad outcomes. So we tested whether in endothelial cells, ApoL1 is hypoxia regulated and which PHD isoform affects them. So we found that, in fact, PHD3 inhibition induces the expression of ApoL1 in endothelial cells, and it synergizes with interferon gamma to further upregulate it. And if you overexpress, PHD3, you can suppress ApoL1, in endothelial cells. So, when we take, all this data together, the conclusion is that there is really a need for more specific therapies in terms of PHD inhibition. The current inhibitors inhibit all PHD isoforms when you only need PHD2 inhibition to accomplish EPO induction. And so that basically calls a need for, more post-approval, monitoring of patients, like, of African ancestry that have ApoL1 to see how they do in terms of different clinical outcomes. So with that, I will stop with my acknowledgement and more important slide. So most of the work that I presented, the JCI paper was, work that, with very significant contributions by my postdoc, Ratnakar Tiwari. who recently moved to another position and has been an amazing, postdoc scientist. The PhD3 work is a work, shared by Ratnakar Tiwari and Rasni Sharma. We are very excited to continue the work, on MCT4 inhibition. There is another, a research program that looks on other metabolic pathways like tryptophan metabolism that I have not had, the chance to highlight today. So, next time. And of course, I would like to acknowledge the support, from our collaborators, Nav Santel, Sue, Quagen, Ed Thorpe, our funding mechanisms, and, we'll stop here and take, any questions you have. Yes.